Thank you all for staying. I know it's been a long day, and I want to thank the organizers, the Broad. I've never been here before, so it's really quite a dream for me to stand up here. I remember watching Tim Yu and Julia Vitarello stand up here and have a conversation, and I was like, one day that'll be Angelman syndrome, and here I am, and I have Tim in the audience, which is such an honor. Melinda, thank you so much for the invitation, the Tremere Institute. It's really a true honor. So I stand here today really to talk about one thing, and I hope I can bring everything together because the stories today, Hillary and Sonia certainly are really the center of where I'm at. And when we think about everybody that spoke today, I think it all centers with one North Star, and that's why are we here. And that's why I'm here. So I'm gonna walk you through a journey. That journey is something that you've heard a little bit about today, but mine's a little different. I am from New York and I have 15 minutes, so I'm gonna do this at New York speed very, very quickly. But one thing I want you to ask yourselves, because I think many of you in the audience are parents, and I just want you to ask yourself, not what would you do for your kid, but actually what wouldn't you do for your kid? Because that's the decision we face the day we get a diagnosis for our child. And so I was already introduced. I have a lot of roles that I play. I wear a lot of hats, and I'm going to walk you through all of those hats that I wear. Of course, I'm the Chief Science Officer at the Foundation. I co-direct a pre-competitive consortium for endpoints and biomarkers to ensure drug development is fair, and there's an even playing field amongst all industry members that are working toward a common reason to develop therapies for a patient population. I'm the Chief Development Officer at our accelerator called AS2 Bio. I'm a strategic advisor to many neurodevelopmental disorder genetic companies, one being Therapeutics, which is a true honor of mine. I'm the director of interventional endoscopy services, so I work in medical device development in animals with naturally occurring diseases. That's where my career started. And I still do that every day because that is a hobby of mine that I love, and it used to feel complicated and now it feels somewhat brainless. To be able to put stents in dogs and cats for ureteral obstructions and biliary obstructions. But the most important job that I have is that I'm the mother to 3 beautiful little girls. And my middle daughter, who's pictured here, Quincy, she's 11 years old and she lives with a de novo mutation called Angelman syndrome. So I'm gonna walk you through the patient journey, which is where we start. That's where Hillary almost ended. That's where I started. The clinician journey is what it takes as a clinician to understand drug development and understand how to solve clinical unmet needs, which is where I have been in veterinary medicine my whole career. A community journey, because I advocate for everyone in this community, of which there are 500,000 people potentially in the world living with Angelman syndrome. So I don't just advocate for that beautiful little girl, but I advocate for every single one of the boys and girls and adults that are living with Angelman syndrome. And then the industry journey that I wound up taking, not because I wanted to, because I had to, and the holistic journey because we're not going to be successful unless we think about every one of the hats that we wear. And so where I started was I got a phone call. My daughter was 5 months old, and on the phone, Dr. Berent, I have catastrophic news for you. That's where I started. I was sitting in a coffee shop writing a paper. I didn't hear exactly what he said other than catastrophic. And I said, what? And he said, your daughter has Angelman syndrome. And I said, how do you spell that? I didn't even know what this man said. Angelman, like what does that even mean? She was developmentally delayed. Everyone told me she was totally normal at developing at her own speed. Maybe she has a little bit of cerebral palsy. A little bit, just like that much. That's what I got when my daughter was diagnosed. And I Googled it, which is exactly what every parent should never do, particularly not a clinician scientist. And I opened the first thing that popped up, which was Wikipedia. And I read all the things that Quincy will never be able to do. She will never walk, she will never talk. She will never live an independent life. She will likely not have friends, she will not get married, she will not go to college. All the things you dream about for your newborn little girl were just destroyed in a minute. And then I thought to myself, well, what will she be able to do? What is the prognosis for survival? And how do I give this child the best life that she's capable of? Because that's what she deserves. And all of this went through my mind in literally 30 seconds as I'm Googling Angelman syndrome. And really what was so clear is that I had to advocate for Quincy because the message that this was delivered by this doctor was so clearly not someone who can advocate for her that I had to do it. And I really had to learn everything about this if I was going to change the trajectory of what I was reading on Wikipedia. And then I fell to the floor. And I said, I can't do this, and quite frankly, I don't want to at all. I worked my whole life to be a veterinarian. That's all I wanted to be since I was 8. I was exactly where I wanted to be in my career. Peer-reviewed, published hundreds of articles, wrote my own textbook. It was exactly where I wanted to be, developed the first interventional radiology service in the world for veterinary medicine. That's what I wanted to do, and that's what I was doing. I was in New York City. I was finally in a place I wanted to be. My career was everything to me. Frankly, I didn't even want kids. But then I got married, and it's like the next thing you do. So I had kids. And here we are. I have this child. And what do I do with her? And I really didn't want it. And I'm going to be honest, because that's the hard truth of it. Most of you are here because you wanted to be here, and you worked really hard to get here. I worked really hard, too. And I was suddenly in a place on the other side of that line, Hillary, where I didn't want to be. What kind of mother thinks like this? That's true. I thought this way. I fell to the floor. I didn't know how I was going to handle this as a family. I didn't know how I was going to handle this as the mother to her older sister. And I was soon to be pregnant with her younger sister. How was I going to be a good wife? How was I going to manage my career? I was not ready to give that up. All in about 3 minutes, this is what happened to me. I really live my life as an interventional radiologist when there's a medical problem that things are either possible or impossible to fix, and there's no in between. And when you have an obstruction, you solve it. You unobstruct it. You open up closed tubes and you close open tubes. That's the way we practice medicine. How is this any different? She was broken and we needed to fix her. And I was naive enough to believe it was easier to fix a single gene disorder than a feline ureter. And so I decided that I would fix her. And I also know that knowledge is power, and that things are either possible or impossible, and there's no in between. And I needed to figure out a way to make this possible. And the only way to do that was to solve the problem that no one could tell me was unsolvable. They just hadn't yet solved it. And so I jumped in head first. I realized I had to be brave, and I had to be smart, and I had to figure this out. I called every senior author I could on every publication I read in PubMed, of which thank God for them, because there were thousands of articles on Angelman syndrome. Why? Because it's a very unique disorder that is an imprinted gene, and you can actually fix it in every single way with genetic medicines. So a lot of people were working on Angelman syndrome. Most of them called me back. Most of them wanted to talk about Angelman syndrome. And I couldn't believe it, because I heard rare, and I was like, she's the only one in the world with this. But what was so clear, and I always say this, was that it was a great decade to be a mouse with Angelman syndrome. They were cured. But it was not the right decade to be Quincy. And that was also the unfortunate part that was very clear. Translational science is generally very much outside the wheelhouse of the most brilliant minds in the world. They go into neuroscience because they want to discover things, and then they find shiny objects that ask more questions, and they follow the questions to answer the questions. But sometimes it's a hamster wheel, and people don't realize that there are humans waiting on the other side, waiting to move forward toward human translation. But people get stuck in, like, what do I need to get the next NIH grant? I need to answer this biological question. And that pauses drug development in a really enormous way. Understanding biology to the point in which you have to, but not to slow progression of what could be transformative in people's lives. And that's where I think a lot of people get lost, when you don't balance medicine and science. And so that was very clear. And so I realized I could not wait for a treatment for Quincy. And if no one was going to do it, what can I do to help develop one for her? And so I learned everything I could about genetics, of which I hated in veterinary school, and neurology, which I hated more in veterinary school. But I learned everything I could about Angelman syndrome. It's on the 15th chromosome. It's an imprinted gene. It's paternally silenced with this long non-coding RNA called the UBE3A antisense transcript that is only expressed on the paternal allele in neurons. Only in neurons. All of us are paternally silenced, but we all are expressing the maternal allele. right here, of a gene called UBE3A. But in Angelman syndrome, it is silenced, but the UBE3A gene is either deleted or mutated. So they have no UBE3A in neurons, but they are haploinsufficient peripherally, and they're completely normal peripherally. This is a significant neurologic disorder of neurons only, and they're totally null in neurons. So what we know is 50% could go a long way, because we all only have 50%, and peripherally is not our target. Neurons are our target. And so with that single gene disorder comes a host of challenges. This is a neurodevelopmental disorder that is non-degenerative, but it is highly debilitating. And ultimately, the most important thing to recognize in the symptoms, like every other neurodevelopmental disorder, is that there's a universal lack of speech, which is devastating. Some of these kids sleep 2 to 3 hours a night. They can have debilitating seizures, and the list goes on. Other thing that's most important is that they can never live an independent life. They need adult support for the rest of their life, but they will live a normal length of life, not a normal quality of life. And we can't forget the impact that these diseases have on families. Imagine a lack of sleep. These kids sleep 2 or 3 hours a night for their whole life, not just in infancy, until they're 50 or 60 years old. What do the parents do with that? It's devastating to live a life with a full-time job where you don't sleep. And these are the unbelievable symptoms that families have because of the disease of their child. And we can't forget about that. The first paper that actually changed my world was the paper on somatic mosaicism and Angelman syndrome. There's a small percentage of the population, about 3%, that have mosaicism, and they have generally 1 to 10% of UBE3 expression on the maternal allele. And because of that, what we can see symptomatically is if they have 1 to 5%, they can speak. If they have 20%, they speak in full sentences with no seizures, and they can walk normally. And if they have over 40%, they're not diagnosed, and it's probably because they're normal. So what we know is if my daughter got 1 to 5% of UB3 expression, it would be a different child than I live with today. So a little bit can go a long way, and the enemy of good is better. If we try to wait for 80% of neurons to be transduced to think we have a therapy in a disease like this, we're going to be waiting the rest of our lives. Minimally important differences and a meaningful change is 1 to 5% for me. And that's what drug developers don't realize. What is important and meaningful to families? And so I had to start taking this community journey. I wound up joining the Foundation for Angelman Syndrome Therapeutics about a year after her diagnosis, when I even knew there were foundations for things like this, like who even knew? And I became their chief science officer, and I was lucky enough to be able to be surrounded by brilliant scientists who were working night and day on Angelman syndrome. What was clear is they didn't know how to develop a drug, but what was also clear is they were brilliant, and they could do it if they had the guidance. And so really we needed to figure out how to scientifically de-risk every approach for Angelman, and we needed to come up with a plan and the financial budget to be able to pay for that plan. How do we get from mouse candidate to human candidate? How do we get from human candidate to first in human? And we had to break it down in bite sizes. We needed go and no-go decisions for everything. And we had a budget for 24 months of $5.8 million to get 6 therapeutic candidates to human candidate, not to first in human. We raised that money in about 6 weeks, and that changed everything. And we committed to do that in 24 months. Before you know it, we can replace the missing gene, we can replace the missing protein with hematopoietic stem cell gene therapy, AAV gene replacement therapy, enzyme replacement therapy. We can unsilence the silent mechanism with an antisense oligonucleotide, with an siRNA, with an miRNA, with a CRISPR gene editor. We can do all of it. We can think about the downstream targets as a synaptopathy, so small molecules might actually have a clinical benefit. But while doing that, we had to be prepared for how are we going to run these trials? What's meaningful? What's measurable? How do we design these trials in a way that levels the playing field for all of these technologies? And so we did that quickly. But as the science progressed, this is an Angelman syndrome mouse on the left, and this is that same mouse after treatment with hematopoietic stem cell ex vivo autologous gene therapy. Okay, it's a great decade to be a mouse with Angelman syndrome, but it's time to move forward. Prospect of benefit and proof of concept was very, very clear. This is a CRISPR gene editor with paternal unsilencing. Wild type on the left, Angelman untreated in the middle, and a gene-edited mouse treated as an adult mouse with Angelman syndrome that actually outperformed the wild type mouse. So what does this tell me? Maybe age doesn't matter. Everyone said if I didn't do this at birth, there was no chance in a neurodevelopmental disorder that we can change the trajectory of the disease. Well, maybe that's not true. So we did a lot of mouse work. And you could see every single modality in the mouse was looked at, from reinstating the gene to delivering it, knocking it down, every which way. And this is the heat map. If you reinstate, it tells you if you do it in an adult, you have little chance of benefit. If you do it immediately postnatally or prenatally, you have a better chance for benefit. Destroyed the field in thinking, oh my gosh, well, these kids are already born. All right, well, what happens if you add an AAV gene therapy to an adult? What if you do an HSC? What if you do an ASO or a CRISPR? What you can see is a lot of that red went away. Therapeutic translation is very different than creating a Cray-Lox model where you give it tamoxifen to turn on a gene. That is not what happens in the real world. That is not what our kids are living with. So when you start to see what therapies can do, you start to change your mindset. And so ultimately, we moved forward, we moved fast. We have a 20-year natural history study showing the natural history trajectory of biomarkers and outcome measures in this disease. We did everything we were supposed to do. We created a pre-competitive consortium engaging with the FDA and the EMA. We created a master protocol for anybody to be able to run their clinical trial based on these endpoints and biomarkers that we paid for and we share freely because we wanted to even the playing field for everyone. And then in 2017, everything changed. One of those 6 programs discovered an antisense oligo that knocked down the antisense and upregulated the paternal allele in human neurons, and the mouse model showed rescue a phenotype in an adult. And we were able to say, well, if no one else is going to do this at our timeline, we're going to do it ourselves. And we started a company called Genetics Biotherapeutics. Long story short, we moved mountains. We got this from human candidate to clearing an IND in 17 months. We started our phase 1/2 on the backs of lemonade stands and golf tournaments, angel investors, and ultimately wound up partnering this program with Ultragenyx Pharmaceuticals for them to be able to take an option where we ran the phase 1/2 and they could acquire with positive clinical data. And that's exactly what they did. We had positive clinical data. And this is just that natural history trajectory. Following on was Ionis Pharmaceuticals had an ASO for Angelman, and Roche had an ASO for Angelman. And it went on. And what you can see here is this is natural history in communication. This is after treatment. This is natural history in cognition. And this is after treatment. And Ionis found exactly the same thing. An enormous natural history comparator benefit compared to the 20-year natural history study. And so what does that heat map mean? Well, this is the ASO. What happens when you have a gene therapy that's now in a phase 1/2 clinical trial, an HSC that's about to be in a clinical trial, or a CRISPR gene editor that's hopefully a soon follow? So what we know is there's a lot of green compared to that red. And if the ASO has transformed the lives of these kids, imagine what genetic targeted therapies that's permanent might do for these kids. And that's where we have to think. So we have to realize drug development is really hard. It takes a very long time and it's very expensive, but it doesn't have to. It does that because we are in an environment where we're used to developing drugs for common diseases, not for rare diseases. It should not have to cost us $275 million to develop an ASO. That just should not be the case in rare disease. So our foundation implants money like this right into the de-risking in the human drugs so that we know it will not be shelved or paused based on what we see in the market right now. When Quincy was diagnosed, everything was academic, 5 programs. After that $5.8 million investment, we had 15 programs. That went to 34 until the stock market crashed in 2022. And after Vinay Prasad shared some news recently, things went down in biotech. And that 34 went to 24. But where we are today is that we have 30 programs in development, most of which are being developed by our foundation. Not because we want to, because we have to. Because no one else is doing it at a pace that matters to our community. And so we started an accelerator to be able to do that. And so what's really important is that traditional drug development is really failing rare disease communities. And we can't let that happen. And it's because it doesn't make enough money based on the regulatory environment that we live in right now. And that has to change. And I think that's what everyone is trying to do. But to meet that gap, our foundation had to fill it. And that's unfortunate because we have to take care of our kids. So we started an accelerator called AS2 Bio, where we took venture philanthropy from patient advocacy and we created this disruptive environment in which we were just going to do it because we could. And that's exactly what happened. We wound up getting a $40 million NIH grant to it as part of the SCGE as well, in collaboration with Yale and Rush University, to advance a CRISPR gene editor that's non-viral delivered. We got $6 million from CIRM to develop our HSC program. And we launched our AAV gene therapy program just this year with our first in-human clinical trial for AAV. And so we dosed our first patient, and this was our announcement there. And so here's where our pipeline is now. We just closed the phase 3 with Ultragenyx. Our AAV gene therapy is in the clinic. Our CRISPR and our HSC is hopefully getting to the clinic very soon, and we have many programs following along. We've created a platform for one disease that is technology agnostic, because what's so clear is that drug development is hard, but what I can promise you is living with rare disease is a heck of a lot harder. That I assure you. It's hard to rely on other parties to have you as a priority, but ultimately, they can change overnight, but I can't. I go to bed every night and I tell my daughter, tomorrow's gonna be a better day. And that's not gonna change until tomorrow is a better day. It does change for industry because they don't answer to the patient every day. They answer to their investors, unfortunately. And that's just the way it is, not because they want to, but because that's the way life goes. So we'll continue to push all shots on goal. We need to work together to improve efficiencies and serve as an example for all others. But remember, things are either possible or impossible, and there's no in between. And so far, things look possible. And that's just one reason out of 500,000. That's just one. And there are thousands of rare diseases that can't have to do what we did. It's so much work. There has to be a better way. Thank you.
Thank you for sharing your story. It's incredibly impactful. How were you able to speed up and bring the cost down from that very familiar timeline and cost perspective to the one you are today?
Yeah, I wouldn't say we brought the cost down. I would say we sped it up because when you have a team of 5 developing a drug, you don't have any red tape about getting decisions with different groups and different hierarchies. We were able to pivot and make decisions based on the people that surrounded us. So we had an incredible team of advisors. And we had 5 main people working on the program, but we had an incredible team of advisors where we were able to make decisions within hours if we needed to pivot on something. We raised the money and we had angel investors in our foundation backing us, but it costs the same as it would cost any pharmaceutical company. The reason it costs a little less is because we were faster. And I think we were faster because people believed in our mission. The CROs believed in our mission. They found us monkeys the minute we needed them. We needed to add another arm to the study. We wanted to follow a study out a little bit longer. People moved mountains because they saw what we were doing is gonna have such direct impact that we weren't stuck based on what the board was thinking is gonna be most investable or most financially impactful. It was more about what's gonna be best for the patients. And that is one and the same, it should be, right? But ultimately, when the board is truly deeply invested in the patients because they're parents, they can make, and they're smart parents, not like I'll just do anything and throw everything against the wall. These are parents that can make go decisions as easily as they can make no-go decisions, and that's very important. So we were able to pivot on things that I think a lot of companies that I see, that I help support other companies, really struggle with. And part of that is because our bottom line is the North Star. How are we gonna get this to patients? Because the true experiment is the human one. And we can talk to the agency about, can we avoid this? Can we avoid that? Do we really need to do 2 species talks? Things like this. And we were able to get a lot of feedback where we pushed the envelope because we weren't afraid for people to say no. Where I think in industry, people are afraid. They just kind of go with the norm because they're afraid for people to say no, and they may go on clinical hold. Oh my God, that's gonna be horrible. Where we weren't afraid to say, we can't afford to do that, so can we try something different? And we got the feedback, yeah, you can. So I think that's what helped.
It's a real inspiration. Thank you.
Thank you.
I'm gonna start with a comment, Allyson, which is that one of the things that she hasn't told you about the work that you've described and that you've led at FAST is one of the textbook examples that I think many people will be studying for a long time. And one of the things that Allyson didn't share with you all is the InSync program, which is helping disseminate the learnings that they've gathered through the Angelman's community in running this remarkable process to many other rare disease communities. And I just thought I'd invite you, for folks who aren't able to attend InSync on a regular basis, to share with us some of the learnings that you find most important to those rare disease communities. Maybe some of the things that you've learned not to do through FAST's efforts over these last couple of years.
Yeah, thanks, Tim. Thanks for that. Yeah, we decided, you can only have impact on one disease, then you only have impact on one disease. If you only have impact on one child, it's only one child, to your point, Hillary. So we started this thing called InSync, the International Angelman Syndrome Research Council, which is a brain trust of drug development in neurodevelopmental disorders, because there's nothing we do that should be kept in a silo. Everything needs to be shared. All of our data is shared. Everything we learn is shared. I am such an open book about things I shouldn't be at times, but it's because why should anybody, if we go faster than most industry members, why should anyone go slower than us? They should go faster than us. We build upon each other. And I want the next generation of diseases that are being diagnosed today to be able to go faster than we did, and they can't if they don't take the learnings that we had. So I'm very open about everything we did. And I think the biggest learning I've had has been to not listen to people when they tell me that you can't do it. And I always thought that way, but sometimes you look at industry and you look at some of these really big scientists and they're intimidating. A lot of people look at me as just a mom. And it's intimidating to walk in a room and people are like, nice try, you're never gonna be able to do that. And then when you do it, and you're just like, I want to go back and be like, do you remember when you told me, but I'm just not that person? I'm not that person, but I think the learning for me is never say never, and ultimately surround yourselves with people that are smarter than you. I am very clear on what I don't know, but I'm also definitive about what I do know. And so it's very easy for me to make decisions. And I think a lot of rare disease communities waffle because they don't have the knowledge, which means they need to surround themselves with people that do. And that's very important. You have to make smart decisions, not emotional decisions. And that's sometimes hard as a parent. And I do think that that's the key to success.
So we've got nodal biology, we've got nodal technology, and nodal processes that you are sharing with all of us. So
yes.
Thank you.
Thank you.
Thanks, Allyson. Really compelling journey, and what you've done is really trailblazed for many other companies. I think it's fascinating with the partnership with Ultragenyx. You don't have the insight as what they're doing from a prioritization in company and how they're spending and doing things. So if you, given the ability to reflect on making that decision, are they moving as fast as you want? Are there lessons learned that you can kind of share with the people here on kind of what you've learned through that partnership, and what would you do different?
That's a great question. I would say we had a lot of takers, of people that wanted to partner this program. We were really so humbled by how many people believed in the data they saw, that they wanted to actually partner this program. So it wasn't, we just went with Ultragenyx because. We really thought it carefully. We had VC interest, we had investor interest, we could have taken it all the way through ourselves, and we had other pharma interest. We purposefully chose Ultragenyx, and the main reason we did was because we felt if something went wrong, they would try to figure it out, and believed in the patient centricity of the company. Not to say other companies don't have it, they particularly had it. And that is 100% true, because we had a very severe adverse event, and we were running the clinical trial as genetics. We ran the phase 1/2, they acquired after. So they optioned in, but we were the sponsor. And we had a very severe adverse event with the ASO. And any other company would have killed it. And they did not, because the effect that they were seeing in the kids was so profound, even though there was this adverse event, that they felt that they can mitigate it with dose adjustments and really think it through and modify and pivot as needed. They didn't just drop the disease, they didn't drop the candidate, they sat down and thought it through and figured it out. And they enrolled 129 patients in their phase 3 in 6.5 months. That's unheard of. So I don't know that another company could have gone faster. Of course, I believe they probably could have, like, put me in there and I'll make it go a little faster. But I think they're a big company, 1,500 employees, well, now a little bit less, unfortunately. But I was mind blown by how committed they were to Angelman. And now we've seen other industry members, Ionis, and now Oak Hill is taking on the other ASO. They're so committed to Angelman. And I do think those companies are out there, but it is hard to know. And you have to have clawback rights. You have to ensure that you have milestones and deliverables that are what you think are logical in your deal, in order to be sure you could take it back at any time if they're not meeting your requirements. And we had really smart lawyers that helped to do that, because there's no way we're letting this get shelved. Even if it meant we had to change candidates for whatever reason, the technology itself we strongly believed in. And now we're looking to partner our AAV gene therapy and then eventually the other programs as well, and we're gonna have the same criteria as we do that. We can't let anyone shelve it, which is why we like to take it through human, because then no one can argue good human data. They can argue good monkey data, but they can't argue against good human data. So it's hard. And sometimes you're like overwhelmed with like money, because a lot of people offered more money. But it wasn't about that. It was about the partnership.